Take it away, fella. Okay, so as you can see, this is our cell membrane. I'm Kerr, and my partner is Jalen Reeser. So this is our cell membrane. Okay, it is a phospholipid bilayer that is basically a head and two tails. The head is polar, and the two tails are nonpolar. No water should be near or in the nonpolar region. And then the polar side, the head, is what lets things in and out. So, yeah. So the surface markers are the Q-tips in this diagram, because all they do is they just mark the surface, and they're kind of linked together, at least a little bit. The channel proteins are next. Those are you can't really see them, but they're the straws in between each yellow thing and in between each cotton ball. Okay, and then that goes in and out from the side. And then the receptor proteins and carbo chains. The receptor proteins are external and used for communication. Those would be cotton balls, or no, they would be the bacterial-looking balls. They're just the fuzzy ones. And then the carbohydrate chains make sugar, but they aren't used for anything else. They're linked to other things. There's a cytoskeleton filaments, which could also be represented by the bacteria, which give it strength and flexibility. And then there are membrane proteins, which could be the cotton balls. So those just give it protein, and then they would be the yellow. They would be the Play-Doh, because they're inside and they maintain shape and they have enzymes. And then the glycoproteins are they make protein, and they're used for they're protein and carbohydrates, and they're used for recognition. So and then the glycolipids are phosphate and carbohydrates, and those are used for protection. They protect the membrane. So the glycoproteins recognize what goes in and out and lets that happen. And then the glycolipids, those would also be the Q-tips. And then don't do that. Pause it, you moron. I sent it again. Okay, so the membrane uses size, shape, and recognition to decide what comes in and out. And in our diagram, those would be enzymes traveling through from top to bottom. So size, shape, and recognition. And the recognition, again, is left to the glycoproteins. So thanks for listening.
Peace out.
Yeah, peace.